Hello and welcome to our lab number 6, and that is enzyme properties. We are going to look at the efficiency of enzymes as a catalyst who accelerate the rate of the reaction. We're going to be looking at what are the properties that enable the enzyme to perform this action at a high capacity. And we're also going to be discussing what are the disturbances that could happen to this enzyme that would affect the reaction rate. So before going into the lab, let's go to our uh, slides that we posted about the topics that we have discussed in class. And let's go to the slide that is number, I believe, 70. Okay, so let's begin here. So we have to always understand that we have macromolecules. These macromolecules are either sugars, they're either lipids, proteins, or they are nucleic acids. So we have discussed the lipids and we have discussed also the nucleic acids and we have discussed the carbohydrates. And now we're discussing enzymes, and enzymes are proteins. So proteins come in a primary structure, and based on hydrogen bonding, they fold into secondary structure. And following that, more extensive hydrogen bonding, in addition to hydrophobic interactions, lead the secondary structure to fold into a tertiary structure. And we have enzymes that are fully functioning at tertiary structure. And some enzymes have more extensive hydrogen bonding, hydrophobic interactions, as well as ionic bonding that would give them quaternary structures. So these enzymes, so not all proteins are enzymes. Some enzymes are structural, some proteins are structural proteins, like the collagen that is in the ligaments of the bones. And there's also keratin that's on your nails. That is not an enzyme. But so not all proteins are enzymes, but all enzymes are proteins. So what do enzymes do? We discussed in class that enzymes are catalysts. Enzymes are catalysts. And catalyst means that they speed up the reaction. The reaction would take forever and they would get you there from the product to from the reactant to the product side faster. So let's look at this reaction occurring so slowly without a catalyst. We have to go up this bump right here, this green, we have to go up this green mountain. So you see that we refer to this as the activation energy. This green barrier is the activation energy. So what the reaction, what happens to the reaction in the presence of the catalyst is that the catalyst only drops and decreases the activation energy. And now we don't have a mountain to climb. We only have a little hill to go over. So obviously the reaction with the catalyst is going to be faster. We discussed in class that these enzymes bind to the reactants on a site known as the active site that was on the quiz. And we discussed that this happens when, we have an enzyme substrate complex. And at the top here, that's your transition state where you're no longer a reactant, but you're still not a product. You are in a transition state. And then we release the product. So this is our active site. This is the enzyme active site, and the substrate or the reactant binds to the active site, and the active site causes some structural rearrangements to the chemical bonds that are in the reactants, and then causes some temporary changes to give us our products. So now let's go back into our lab. So what are we looking at? We are looking at the effects of this very important enzyme that is a form of a catalase. Catalase is a big family of enzymes. This enzyme is known as peroxidase. So we're going to be looking at peroxidase, peroxidase enzyme properties. So peroxidase is going to, we cannot look at peroxidase in isolation. So we have to understand that, as we mentioned in class, we have a superoxide ion. This superoxide ion is a form of a free radical that causes very dangerous oxidative stress to the cell. So the cell is built in with mechanisms, is equipped with mechanisms, with enzymes, and we mentioned SOD, SOD, superoxide dismutase, that mutes this superoxide ion into hydrogen peroxide, H2O2. Hydrogen peroxide then will be taken by a catalase, which is peroxidase. Peroxidase, catalase, same thing. Peroxidase belongs to the family of catalase, and it's going to be converted from hydrogen peroxide into water and oxygen. We mentioned in class, we all know what is the value of water in our body, and that is for blood volume, euvolemia. We don't want our osmolarity to change. We need water for the adhesion, we need water for chemical reactions and metabolism in our body, and oxygen, which is very useful. And this oxygen, we need it for oxidative phosphorylation and for aerobic respiration. Okay, so this is what's supposed to happen to repair this oxidative stress. If this oxidative stress keeps going on, so the normal cell is susceptible to continuous oxidative stress. And these free radicals, like superoxide ion, because it gains, so O2, that is neutral oxygen. That's what we want for oxidative phosphorylation, and that's what we want for aerobic respiration. But with a minus charge, now it gained an extra electron, so that's very dangerous. So now it's no longer neutral oxygen, that's a superoxide anion. If you lose an electron, you're a cation. If you gain an electron, you're an anion. Then these superoxides and these reactive oxidative species will cause oxidative stress that would accumulate in the cell to damage the cell. And with continuous oxidative stress, the cell will be damaged and it will reach a level known as damage beyond salvage. The cell will be damaged beyond repair. So you have to understand that these free radicals are, atoms that gain an extra unpaired electron. So that unpaired electron in the superoxide anion is an example of a free radical. And free radicals increase, so we need antioxidants. So these guys here, the superoxide dismutase, the catalase, the peroxidase, these are antioxidants. So we have to understand that reactive oxidative species are very dangerous. We get reactive oxidative species because of many reasons, because of radiation, because of cell uptake of microbes like infectious microbes. The mitochondria in its reactions give us metabolic waste in the form of reactive oxidative stress. Endoplasmic reticulum with unfolded proteins gives us also a form of stress, reactive oxidative species. And there are many reasons that produce, many causes that produce reactive oxidative stress. So what we're going to do in our lab is that we're going to use this very important Guaiacol or Guaiacol reagent. This Guaiacol reagent is colorless. This is very, very important. So what happens is this is happening here at the level of the purple. So we no longer have superoxide anion. We have the hydrogen peroxide. So we have two reactions that will happen. Hydrogen peroxide will react with peroxidase. And with peroxidase, what peroxidase will do, it will oxidize hydrogen peroxide to give us oxygen and water. Then the second part of the reaction is that's where Guaiacol comes in. Guaiacol is colorless. And what Guaiacol does is if it finds oxygen as a product, the second reaction will be Guaiacol is reduced, it has extra electrons. It will donate those extra electrons to the oxygen and it will bind oxygen. Once Guaiacol, who is colorless and reduced, who has electrons, once Guaiacol, who is colorless and reduced, possessing electrons, binds to oxygen, it will no longer be reduced, it will now be oxidized because it bound to oxygen. It lost the electrons because it gave them to oxygen. And now Guaiacol is no longer colorless, it will turn into amber color or brown color. So if you drop Guaiacol in a reaction with hydrogen peroxide, nothing will happen. If you add Guaiacol to hydrogen, to hydrogen peroxide in water, it will not change color. If you add Guaiacol to peroxidase only, it will not change color. But if you add the three, if you have Guaiacol with hydrogen peroxide and peroxidase, so you know the two reactions will now happen. The first reaction will be peroxidase will oxidize hydrogen peroxide, converting it to water and oxygen. Then Guaiacol will come and it will bind oxygen and it will donate electrons to oxygen. And it will now become, Guaiacol will be changed from being reduced Guaiacol, who's colorless, to oxidized Guaiacol, who is amber color, dark in color, amber color. So now the experiment, we have to look at our materials. Our materials is we can get this very important enzyme known as peroxidase from the beetroot plant. We can also get it from broccoli plant. And what we do is we get a sample, we grind it. We can use a blender or we can crush it down with a grinder. And we can take that extract, add water to it, and we can remove the extract and filter it through chromatography paper and get all that liquid extract out. Then what we'll do is we will do something called blanching. Blanching is we will go and we will take this crushed or grinded or blended beetroot plant or broccoli plant, same thing. And we grind it and we filtered from chromatography through chromatography paper, and we get that solution. We heat it up, we boil it. When we heat it up, all of the other enzymes that will interfere will denature. But peroxidase is actually heat resistant, so it will survive. So now we have eliminated all the other outliers. We have eliminated all the other enzymes that could be in this plant. And you know plants, why do plants have peroxidase? Because them too, they don't want oxidative stress. So it's not only human organisms or animals whose cells are damaged by reactive oxidative species, plants also are damaged by reactive oxidative species. So they do also have a system, an antioxidant system with all of these catalases, just like we do in our body. So we cannot get it from human cells or animal tissue, but we can get it from the plants. That's much cheaper. So we grind the beetroot or the beetroot plant or the turnip plant or broccoli, and we can grind it with a blender. If you don't have a blender, we can crush it down and we can take the extract and add water to it, and we can add buffer. Sometimes alcohol is also added. And what we do is we filter that crushed sample and we filter it, and what comes down will be that colored solution containing hydrogen peroxidase, that enzyme that we need. Then we're gonna boil it. After we boil it, we're gonna add it to cold ice, and that is known as blanching to stabilize. Just after we heated it up, we add ice. And then after we add ice, it's very important that we go to the next step. And the next step is we sometimes in lieu of using a blender or using a crusher like this one, this is a crusher in laboratory, we can centrifuge. We can have a solution that has our beetroot plant, and we can centrifuge it and take the pellet, and the pellet will contain our hydrogen peroxide using a pipette. Then what we will do is we will have to rely on spectrophotometry. And we explain spectrophotometry. You have to know these phases. What happens is we have a light source that illuminates white color, and white color is a mixture of all the colors together. And we will have a lens, and the lens will focus this light from the light source into a prism or a filter, and that filter is going to now separate the color. Roy G. Biv, red, orange, yellow, green, blue, violet. And then we will have our sample. And remember, we mentioned in class that we will only absorb at a wavelength that reflects our identity. So no two samples will absorb at the same wavelength. So we will absorb as much as what is in the sample. And we will record the absorbance. Absorbance is how much was impeded, how much light was impeded. Transmittance is how much light passed through. So our hypothesis must be the more dark the solution is, the darker, the more amber Guaiacol is, that means the more product, the more oxygen was produced. That means the more active hydrogen peroxide is. And then we will have different test tubes with different variables, with different independent variables, like different pH ranges with different temperature ranges. And we will also have a control solution. And we will see that temperature will have an effect on the density, on the concentration of that Guaiacol color, indicating that, for example, if we heat up the test tube, the cuvette, or boil it so much, then that is going to denature a lot of our peroxidase, because that's an enzyme. And then we will not have as much product turnover like oxygen, and therefore not a lot of Guaiacol is going to turn into amber. So that's what we're appreciating. So we're using the spectrophotometer to detect the amount of light that was absorbed, and then we're going to plot our results. So we're going to look at the effect of, for example, on enzyme concentration. If we double the enzyme concentration, if we double hydrogen peroxidase, or if we used a standard amount, or we have the amount, how does that affect the absorbance? So obviously, the more enzyme you use, the more products you're going to have, the more dark, the more amber the color will be, and the higher the absorbance. So the higher the impedance and the less the transmittance. So that shows you if you add more enzyme, the reaction rate will go faster. You will have more product. Also, the effect of a substrate. If you add a lot of substrate, that will also have positive effect. You will have a higher turnover. Obviously, when it comes to the effect of the enzyme concentration, it's a linear relationship. So the more enzyme you add, the more products you're going to end up with. However, there's a transport maximum. You see a ceiling effect here. So if you add a lot of substrate that overwhelm the amount of enzymes, the enzymes are going to say we're fully saturated. We cannot take any more reactants. So then the reaction rate is going to stop no matter how much substrate you add. So in the beginning, the more substrate you add, the faster the reaction rate, and the more products you're going to have, and the more the absorbance. But then you're going to reach a level where you're going to hit transport maximum. And that is also like in diabetes. Diabetes, in their urinalysis, you will see there's a lot of sugar. And the reason for that is because, in the kidney, there is these glucose channels that reabsorb sugar from the filtrate back into the blood have been overwhelmed because there's so much sugar in the body of a diabetic patient, unfortunately. So what happens now is these protein channels, who are also enzymes, have been saturated. So this is something that we're going to appreciate in this lab, that is not like the more enzyme you add, the faster you'll go until you run out of substrate. In the beginning, the more substrate you add, the reaction will go faster until you're going to hit a point where now we're going to hit a transport maximum or a ceiling effect. And that shows us that at particular pHs, so enzymes have an optimal pH that they will operate at, an optimal temperature, also optimal salinity. So that shows you that it's not like, oh, the higher the temperature. Obviously, when you increase the temperature, the reaction rate goes faster, you see? because temperature is the average kinetic energy. But the problem is you're going to reach a point where you heat things up so high, that's going to be a problem. You're going to denature this globular enzyme and the pH also. So we have enzymes that work in the stomach at a very acidic pH, like your trypsin. This works at the stomach where the pH is 1. And then you have in your mouth where the pH is 7, you have amylase. That's another enzyme that works at a different pH. So not all the enzymes work at the same pH. They have an optimal pH that they operate at best capacity at, and they have an optimal temperature. And then we're going to be discussing the effects of substrate concentration or enzyme activity. So the more substrate you add initially, things will go really fast. You will have a lot of product, reaction will go really fast. But then you're going to reach a ceiling effect with the transport maximum. And then the temperature, the more you heat, the reaction, the temperature is going to speed up the diffusion rate. It's going to speed up the enzyme activity, the reaction rate. But then you're going to heat it up excessively. That's going to denature. It's going to break the hydrogen bonds between the subunits of your enzyme. May even destroy the covalent bonds. And then the effects of the pH, also remember that proteins, as we said in the slides, we said that you see amino acids have an amino terminal and a carboxylic terminal. That's an acidic terminal that donates hydrogen protons, and that's a nitrogenous terminal that gains an additional hydrogen proton. So if you mess up with the pH, you're going to cause excessive protonation and deprotonation, and that's going to destroy our enzyme and impede its function. So we're going to be coming back to assessing our hypothesis. So we have an alternative hypothesis and a null hypothesis. The null hypothesis tells us to appreciate the relationship between our independent and dependent variable. So this predicts, for example, the null hypothesis will say there is no effect on substrate concentration on enzyme activity. No matter how little or how much enzyme substrate you add, the reaction rate will not be affected. Doesn't matter if you increase or decrease the temperature, reaction rate will not be affected. Doesn't matter pH, what do you do to the pH, nothing will be affected. It doesn't matter if the salinity changes, nothing will be affected. So that's a null hypothesis. But our alternate hypothesis obviously is the more substrate we add, the higher our enzyme activity will be, and we will have a lot of product, and that will be assessed by the color, the depth of color, and the density. The more amber the color will be, and that will be actually reported as data by our spectrophotometer. So that's where we start making objective, deductive deductive and inductive reasoning, saying that the temperature also, if you increase it, we need an optimal temperature, and then if we increase it above that, then the enzyme protein will denature. Also the pH, we need an optimal pH, and so on. In the end, we have multiple conclusions, but we'll just say, for example, that the enzyme catalyzed catalyzed reactions occur faster than the same reactions that do not have an enzyme. And enzymes have an optimal pH temperature settings, and they will denature if these optimal pH and temperature settings are disturbed. So we will talk and explain these topics more in class. Thank you very much, and I'll see you in lab.